Hello, my name is Annika, and this is my mitosis and meiosis video. You can see a picture of me right here as the cover photo. And this is relevant, one, because I look really cool as I dressed up as Marty McFly for Halloween, and two, because I'm a human being who has many cells. Therefore, I have undergone both mitosis and meiosis at some point in my life. So now we'll move on to the first phase, which isn't actually a part of mitosis and meiosis, but it happens before, which is interphase. There are three phases within interphase, the S phase, the G1 phase, and the G2 phase. In the S phase, all the DNA in the cell are duplicated. And then in the G2 phase, which occurs right before mitosis, there is still a nuclear envelope around the nucleus, and all the chromosomes now duplicated have not yet been condensed. As you can see in the picture, you can't see the individual chromosomes yet. And also you can see in the top that two centrosomes have formed, and these will later spread apart to become the mitotic spindle. Moving on to the first phase of both mitosis and meiosis is prophase. In prophase, the chromosomes condense and they become tightly coiled. So now you can see each individual chromosome, and the centrosomes start to move from the center to opposite sides of the cell, and this will become the mitotic spindle. The mitotic spindle is formed from microtubules that arise from centrosomes. And there are two main microtubule types in the mitotic spindle: kinetochore microtubules, which attach to the chromosome, and non-kinetochore microtubules, which don't attach to the chromosome, but they overlap with another non-kinetochore microtubule. For prophase in mitosis, you can see that the homologous chromosomes are not attached. However, in prophase 1, in meiosis, you can see that the homologous chromosomes are attached. And this is because in meiosis, crossing over occurs in prophase 1. Crossing over is the exchange of genetic material between the maternal and paternal chromosomes. So, for example, in this photo, the blue maternal chromosome exchanges a piece of DNA with the yellow paternal chromosome. Also note that in meiosis, when the two chromosomes are attached together, this is called a tetrad, because there are now two sets of sister chromatids together, so there are 4 chromatids attached together in a tetrad. The next phase of mitosis and meiosis 1 is metaphase, or metaphase 1. In metaphase, you can see the centrosomes are now at opposite poles of the cell, and the chromosomes have all arrived at the metaphase plate, so they are all lined up in the middle of the cell, and one chromatid is closer to a pole, so each chromatid is lined up with a pole, so they're all in the same orientation. The kinetochore microtubules are attached to the chromosomes, and the non-kinetochore microtubules are on the outside and they overlap with each other. In mitosis metaphase, the chromosomes aren't stuck together, so the homologous chromosomes aren't together, so they line up independently, forming a single line. However, in metaphase 1, in meiosis 1, the homologous chromosomes are stuck together in a tetrad, so they line up as tetrads. Next, we have anaphase. In anaphase, the kinetochore microtubules are shortening, which eventually causes the chromosomes to separate. So in mitosis, the sister chromatids are pulled apart. So one chromatid is pulled towards each pole due to the shortening of the kinetochore microtubules. As the non-kinetochore microtubules lengthen, the cell elongates. So by the end of anaphase, the two ends of the cells have equivalent collections of chromosomes. In meiosis 1, in anaphase 1, the tetrad is pulled apart when the kinetochore microtubules shorten. So the sister chromatids remain together, but the homologous chromosomes are pulled apart to separate poles of the cell. So then one chromosome containing both maternal and paternal genetic material due to the crossing over in prophase are pulled to each side of the cell. In telophase and cytokinesis, the nuclear envelope reforms around the chromosomes and the cytoplasm divides, which is called cleaving. And it converts the elongated cell into two separate daughter cells. Telophase and cytokinesis marks the end of mitosis. So now at this point in mitosis, we have two diploid daughter cells that are genetically identical to both each other and the parent cell. At the end of telophase 1 and cytokinesis in meiosis, now this is the end of meiosis 1, and we started with a diploid cell, but now we have two haploid cells, each with one chromosome, and the daughter cells are genetically different to the parent cell. Now that meiosis 1 is over, meiosis 2 can begin. Meiosis 2 is very similar mechanistically to mitosis. And it is important to note that in between meiosis 1 and meiosis 2, the chromosomes do not duplicate. We are left with the same amount of chromosomes. We still have two haploid daughter cells that came from meiosis 1. In prophase 2, we see spindles start to form and the centrosomes start to move to either end of the cell, and the chromosomes are condensed with no homologous pairs attached. Instead of having a tetrad like we did in prophase 1, we now just have a chromosome with two chromatids attached together in each daughter cell. In metaphase 2, the chromosomes line up at the midline of the cell, and because of crossing over in prophase 1, the chromosomes are not genetically identical. And we also see that the kinetochore microtubules are attached to the chromosome and the non-kinetochore microtubules are overlapping with each other. In anaphase 2, the shortening of the kinetochore microtubules cause the sister chromatids of each individual chromosome to separate, and the non-kinetochore microtubules overlap and push against each other, helping to elongate the cell. So each sister chromatid is now moving, they're moving to opposite sides of the cell. Finally, we have telophase 2 and cytokinesis. So the nuclear envelope reforms around the chromosome and the cytoplasm divides. leaving us with 4 haploid daughter cells from a single parent cell. So at the end of meiosis 2, we have 4 daughter cells that are genetically different from each other and the parent cell due to the crossing over, and they also each have half of the number of chromosomes compared to the diploid parent cell. Thank you for listening to my very long video about mitosis and meiosis. I hope you enjoyed it.